Hello, this is Chambisino Pharmacosis YouTube channel, where we make your life easier with the subject of pharmacology in both clinical practice and examination point of view. So in the previous session, we have covered the introduction to the general pharmacology, where different aspect is already covered. So this is the second session where we are going to discuss about the pharmacokinetics. Mainly we will focus on the introduction to the pharmacokinetics and the different method for the transportation of the drug across the biological membrane. So in this pharmacokinetics, as the branch of the pharmacology, we say like pharmacology, this is the study or the science of the drug. So it has two main branches, which is pharmacokinetics and pharmacodynamics. So for today, we are focusing on the pharmacokinetics, which is what the body does to the drug, or it involves the quantitative study of the drug movement in, through, and out of the body. So in this case, we are going to focus mainly on the pharmacokinetic parameter, which is the absorption, distribution, biotransformation, metabolism, and excretion. When we say pharmacokinetics as what the body does to the drug, it is the body that absorbs the drugs. It is the body that distributes the drug, and it is the body that involved in metabolism or biotransformation of the drug, and it is the body that excretes the drug. That's why pharmacokinetics, it is the effect of the body to the drug. So for this case, as you see, the drug, once it is administered, for example, for the drug which is taken orally, first it will be absorbed, means from the site of the administration into the systemic circulation. Then it will distribute into the various tissue, including the tissue for the site for the elimination and even the site for the action. Then later it will undergo metabolism before excretion, because most of the drug that you are taking, they are lipophilic. So first it has to undergo the metabolism, then it will be excreted. So pharmacokinetics, by definition, is the pharmacokinetic is the quantitative study of the drug movement in, through, and out of the body. So for example, if you take the drug orally, consider this is the GIT. If you take the drug orally, for those drugs which it will reach into the stomach, for the drug which is acidic, it is absorption, it will take place into the stomach and reach into the systemic circulation. But in case for the drug which is basic, it will be absorbed into the small intestine, means the jejunum or ileum, and it will reach into the systemic circulation. So for the drug, once it reaches into the systemic circulation, there is some amount of the drug which will be existing in form of the free form, and some of other drug it will be in the bound form. So it is the free form of the drug that will undergo the distribution into the site of action and into the tissue which involved for the storage of the drug. And also, once the drug has provided the action to the site of action, then it will back into the systemic circulation. Then it has to be excreted through the excretory organ, for example, the kidney. But if the drug it is non-polar, which means it is the lipid soluble, kidney will not excrete it, because it is only the polar drug will be excreted in the kidney. For that, it will go back into the systemic circulation, and then it will distribute into the liver, which is the organ responsible for the metabolism. Then it will convert those drug which is lipid soluble into the water soluble drug. Then it will be ready to be excreted via the kidney. So this is the diagram that summarizes the whole pharmacokinetic parameter: absorption, distribution, and metabolism, and excretion as well. So for this case, if the drug is absorbed, it is in form of the solid dosage form, is taken orally. It will undergo the disintegration and dissolution, whereby the drug it will be released from the dosage form. Then the drug it will be in solution form. So the solution form of the drug, it will get absorbed into the systemic circulation. So if you can see, this is now the systemic circulation where the drug it will get absorbed. So some amount of the drug, as we said, that it will remain in a free form, while other drug it will bound with the plasma protein, for example, the albumin or glycoprotein. So this drug which is in free form, it will get distributed into the site of action and the storage site. So once it reaches into the site of action, some amount it will be in the bound form, while another drug it will be in the free form. It is the free form of the drug which involve in inducing the pharmacological effect, while the bound form it will be inactive. As we see, some of the drug will be distributed into the storage site, And when it is in the systemic circulation, the bound form of the drug, it means the drug which is bound with the plasma protein, it will not be able to undergo metabolism, it will not be able to undergo the distribution, and even it will not be excreted. So those free form of the drug, it will be metabolized and it will be excreted through different organs which is responsible for the excretion of the drug. For example, here we have urine, bile, feces, sweat, and the saliva organs. So for this case, as we see the 4 pharmacokinetic parameter, so it is very important to understand the pharmacokinetic parameter, all 4 pharmacokinetic parameter, because understanding the pharmacokinetic parameter, it will help you to know the dose or the route of the drug administration. So which route the drug will be taken is through understanding the pharmacokinetic of a particular drug. It will help you to understand the dose, the onset of action, the duration of action, the peak of action, even the frequency of administration between one dose of the drug to another dose. So pharmacokinetics is very, very important to understand the pharmacokinetic of the drug. So as we see all these 4 pharmacokinetic parameters, it involves the movement of the drug into the body. So once the drug it is moving into the body, it has to cross through the different biological membrane. So here we are going to see how the drug is crossing the biological membrane. So we will see different methods that involve for the drug transportation across the biological membrane. So before understanding those methods, let's see the anatomy and the physiology of the biological membrane. So the biological membrane, actually, it is made up of the phospholipid bilayer, which is, they have hydrophilic part, which is present on the surface of the biological membrane, while the lipophilic part, it is present inside of the biological membrane. Also, it is made up of the intrinsic and extrinsic protein, which is embedded into the cell membrane, but also it is made up of those glycoprotein and the glycolipid, which is attached on the surface of the biological membrane. Also, it contains the cholesterol, which is important to maintain the fluidity of the cell membrane. In case of the thickness, if you see the biological membrane, it has a thickness of about 100 angstrom. So it is very important to understand the anatomy and the physiology of the biological membrane, so that to understand the transportation of the drug across the biological membrane. Now, the following are the drug transport method, which involve the passive transport. It means it involves utilization of energy is not. So for this case, energy is not used. So here we discuss the diffusion and the filtration. Also, we have the carrier-mediated or specialized transport, where the facilitated transport and active transport. So for these 2 methods, there is a special carrier which is required for the transportation of the drug. But for the case of the active transport, it is further categorized into the primary active transport and secondary active transport. The last method that we are going to discuss here, it will be the pinocytosis, which involve the transportation of those huge molecules. So for the drug like insulin, it will be transported through this method of the pinocytosis. So this is the summarized diagram that indicates all methods for the transportation of the drug across the biological membrane: the passive transport, carrier-mediated transport, and the pinocytosis. So in case of the passive transport, as we said that in this, there is no carrier, no transport which is required. It is only the membrane which is used for the transportation of the drug. Energy is not required, and the transportation of the drug, it is along the concentration gradient. So as you can see from this diagram, it indicates both 2 passive transport mechanisms of the drug, which is the passive diffusion and the filtration across the biological membrane. So for the passive transport, it is important for the drug which is lipid in nature. So it will transport from the region of the high concentration to the region of the low concentration. So lipophilicity of the drug is very important for the drug to undergo the passive diffusion. And this is the very common among of the method which involve the transportation of the drug across the biological membrane. Another passive transport mechanism is the filtration, whereby the drug is transported through the intercellular space, through the aqueous pores, which is located into the biological membrane, or through the paracellular space. So the solubility of the drug, most of the drug which is transported through this method, they are non-lipid soluble, they are water soluble. But here, even the lipid-soluble drug can undergo the transportation, because the main limiting factor here, it is the molecular size of the drug. So for the drug to undergo the filtration, its molecular size should be smaller than the aqueous pores. So we are going to discuss it in detail. It is very important in case of the glomerular capillaries. So we have seen already the passive mechanism, which is the diffusion and the filtration. So let's see the diffusion, which is the passive diffusion. This is the most common method for the transportation of the drug. So the drug, it will transport across the biological membrane in direction of the concentration gradient. So here it is the transportation of the drug. It will be from the higher concentration to the low concentration. No energy is needed for this process, as we have seen the passive diffusion, the first mechanism here, whereby the limiting factor is the lipid solubility. So in this case, it is the lipid-soluble drug that can be able to undergo the passive diffusion, transportation from the high concentration to the low concentration across the biological membrane. So as you can see here, apart from the lipid solubility as the limiting factor in case of the passive diffusion, also the ionization of the drug. So most of the drug that we are taking, they are electrolytes. So they are either weak acidic electrolyte or weak basic electrolyte. If you take the drug which is electrolyte, if it is weak acidic, absorption has to take place in the stomach. So because it will undergo ionization, it means the unionized form of the drug, it is the one that will be able to undergo the absorption. So for the acidic drug, because in the stomach there is acid, hydrochloric acid, which is produced, so it will remain unionized. That's why its absorption is taking place in the stomach. For the basic drug, you know, the small intestine has the basic pH, so the drug, it will remain, if it is in basic form, it will remain unionized in the small intestine where the absorption will take place. But the basic principle here, the drug to undergo the passive diffusion, in case it is weak electrolyte, it has to undergo, it has to remain in ionized form. Ionized form will not transport across the biological membrane. So we have seen already the diffusion. Let's see the filtration. As you said, the filtration is the passive transport, but in this case, it will be the transportation from high concentration to the low concentration, but across the paracellular space, which is located, the aqueous pores, which is located in the paracellular space, so through the pores. So the size of the drug is very important. The molecular size of the drug is very important to pass through the aqueous pores. It should be smaller size in comparison with the size of the aqueous pores. So both the lipid-soluble drug and the water-soluble drug, it can pass through the filtration provided that its size is smaller. But this method, it is very common, especially for the drug which is water-soluble. In case of the glomerular capillaries, most of the glomerular capillaries, they have the capillary size of about 40 angstroms. So this is very, very large size, to the extent that most of the drug can be allowed to pass through the aqueous pore, provided that it has a molecular size of about 100 to about 200 Daltons. So they can pass through this the glomerular capillaries, with the exception of the drug, the capillary in the brain. Capillary in the brain, it has a very small size. And they are, apart from being of having small size, there is addition of another layer on the surface, which is called the oligodendrocyte, which restricts the absorption of the drug, which is in form of the water soluble. Only the drug, which is the lipid soluble, will be able to penetrate across the blood-brain barrier. So we have seen those, the passive diffusion and the filtration, and we see that the filtration is very important in case of the glomerular capillaries. As you can see, the blood is coming through the Bowman capsule with the afferent arteriole. So once it reaches into the Bowman capsule, there is a pause which is available where the filtration has to take place. So it is the size that will determine whether the drug will be filtrated or not. So this is very important, especially for the drug which is like the drug which is sugar and those urea. All this drug is get filtered, so it will undergo the filtration. But for this case, for the drug which is in bound form with the protein, it will not be able to pass through because this is a big molecule, so filtration will not be possible. So we have seen the exception for the filtration to take place, especially for the water-soluble drug. We say that because of this blood-brain barrier and the addition of the oligodendrocyte cell or the glial cell, it will restrict the absorption of the water-soluble drug, and there is a tight junction between one cell to another that will limit the absorption of the drug. It is only the water lipid-soluble drug that will be absorbed. So for this case, those water-soluble drugs, including the gentamicin, amikacin, neomycin, and the dopamine, it will not be able to pass through the blood-brain barrier for the reason that there is no paracellular space between one cell to another because of the existence of the tight junction, and it will restrict the passage of this drug because there is addition of this glial cell or oligodendrocyte that restrict the absorption of the drug. So we have seen so far the part of the filtration. Let's see now another method, which is the carrier-mediated or specialized transport, where we're going to see the facilitated diffusion where energy is not used, and the active transport where it involves the utilization of the energy. So in this carrier, mainly it involves the utilization of the carrier molecule or the transporters. But for the carrier in general, this is the specific carrier or transporter which is required for the transportation to take place. So it is very selective, it is saturable, and competitive in this, and especially for the drug which utilizing the same carrier molecule. So competition, it might happen, which can inhibit another drug from passing through the biological membrane. So for this case, as you can see from this diagram, it indicates those carrier-mediated transportation across the biological membrane. We have said that, well, the facilitated diffusion. So the diagram A, it indicates the facilitated diffusion, whereby the drug is passing through the carrier. This is the carrier molecules. You know, this facilitated diffusion, this is the one of the transportation that involve the carrier, which belongs to the superfamily of the solute carrier transporter, which is abbreviated as SLC. So the drug is crossing the biological membrane through this carrier transporter from the region of the high concentration to the low concentration. As you can see from the diagram, the drug is moving from this region of the high concentration to the region of the low concentration through the carrier molecule, which belongs to the superfamily of the solute carrier transport. The B diagram, the diagram B, here it indicates the active transport. So this active transport, as we said, that it is further categorized into the primary active transport and the secondary active transport. For the primary active transport, it involves the utilization of the energy which is obtained from the direct hydrolysis of ATP. See now this, the carrier or the transport which is involved in this mechanism here, it is the transporter which belongs to the superfamily of the ATP binding carrier transport, which is abbreviated as the ABC, ATP binding carrier transport. So it is bind with the ATP, which is hydrolyzed with the ATPase. It releases the energy, and the energy, it will transport the drug from the region of the low concentration to the high concentration, as you can see the diagram for the transportation here. While the diagram C, it indicates the active transport, but in case of the secondary active transport. You see, as you said, like the active transport, we have the primary and the secondary active transport. But in case of the secondary active transport, we said it is further categorized into the symport and antiport. So in case of the symport, this is the diagram C, which indicates the symport, where the drug has to pass through the solute carrier transport with the utilization of a special carrier molecule. So this is the symport, whereby the carrier molecule and the drug, they transport across the biological membrane using on the same direction. So here now, it is very important to know in this diagram which is the drug and which is the substrate. So the diagram, the substrate A here, it indicates the drug, which is in form of the peak here, and then the diagram, the diagram B here, it indicates, or the subset B, it indicates the carrier molecule. So for this case, the carrier molecule, it will be moving from the region of the higher concentration to the low concentration, so down here along the concentration gradient. Once it will be moving as the carrier, not the drug, the carrier from the high concentration, you see this red dot here, it indicates the carrier molecule, which is substrate B. Once it will move across the carrier molecule here, the solute carrier transporter, it will emit energy. The energy that will be released, it will be helpful to transport the drug from the region of the low concentration. As you see, this is the drug, this region is low concentration to the low concentration. So because they are on the same direction, this is called the symport. But for the antiport, the same mechanism, but carrier and the drug, they are moving on opposite direction. So as you said, the subset A, it is the drug. So it is moving from the region of the low concentration here, going to the region of the high concentration. Energy has to be needed, but it is the energy which is released by the substrate B, which acts as the carrier. Once it will be passing through the biological membrane along the concentration gradient from the high concentration to the low concentration, it will emit energy. Then the energy, it will be helpful to carry the drug in the opposite side from the low concentration to the high concentration. Because of the different movement, the carrier is moving in opposite direction with the drug. This is called the antipode. So this active transport is very important for the transportation of the drug, especially the second transport. If you see now the methyl dopa and levodopa drug, it is transportation across the biological membrane with the help of aromatic amino acid. This is the secondary active transport. Also, the absorption of the iron into the small intestine. It involves the transferrin, which acts as a transporter. ATP is used. Also, the absorption of the vitamin B, which in the GIT, which involves the intrinsic factor as the carrier. So here, energy is very important. So you can see here, the transporter as the carrier, we say that they require the carrier or transporter. So the transporter has to bind with the drug, then it will undergo the conformation change. Then the substrate will be carried into another substrate, into another side of the membrane. Once the carrier, it will succeed transporting the drug into another side of the biological membrane to undergo dissociation, release the drug, and go back to the original state, as we have seen in this diagram. So that is all about the carrier transportation of the drug. But if we see it in detail, this carrier mediated transport, as you said, there is transport require the energy. This is the active transport. And the transport which does not require energy, this is the facilitated transport. So for the facilitated transport, which is the diffusion now, this, as we said, it belongs to the superfamily of the solute carrier transporter. And it operates passive without need of the energy for the case of the facilitated diffusion. So it's like the simple diffusion, but in this case, it is the process is very fast in comparison to the simple diffusion. And it is along the concentration gradient from the region of the low concentration to the high concentration. So simple diffusion, or the diffusion transportation in this case is very important, especially for the transportation of the glucose into the muscles and fat by the use of the carrier transport called the GLUT or glucose transporter, which is the GLUT4s. As you have seen here, the transporter is required for the transportation of the drug, but energy is not needed because the transportation is along the concentration gradient from high concentration to the low concentration. So they undergo diffusion through the carrier or through the carrier transporters. So for the active transport, the carrier is needed and the transporter is needed, but in this case, the carrier is the ATP. The carrier is the energy. So if the energy that you used to carry the drug, it is from the ATP direct, this is the primary transport. But if the energy which used to carry the drug from the one region means from the low concentration to the high concentration, it is from another substance apart from the ATP. This is the secondary transport. So active transport in general, it is depend on the energy and it is unidirection. So it is against the concentration gradient from higher concentration, from low concentration to the higher concentration. So this diagram, it indicates the primary active transport. As you see here, it involves the transporter which belongs to the superfamily of the ATP binding carrier transporter, whereby the ATP is direct hydrolyzed, releasing energy that will be helpful for the transportation of the drug from the region of the low concentration to the high concentration. So in this diagram, to understand, the arrow is very important. So you have to see the arrow where the drug is moving and where it is going. So if you see the arrow here, it is from the low concentration going to the back concentration. How this will be possible? It will be possible because the ATP, the energy has to be used. So if you see here, when the energy is required, in this case, when it is obtained direct from the ATP, this is called the primary active transport. But when the energy it is derived from another substance which is undergoing the downward movement or which is moving from the region of high concentration to the low concentration, it releases energy. When that energy is utilized to transport the drug, this is the secondary transport, but based on the direction, it will be classified into the symport or antiport active transport. If the carrier molecule is on the same direction with the drug, that is the symport, but if it is in the opposite direction, that is the antiport. So primary transport, ATP, the energy is obtained from the direct hydrolysis of the ATP, and we say that it belongs to the family of the ATP binding transporter, and it involves in the transportation of the drug with the utilization of the ATP. So the good example here, in case of the primary active transport, it is the P-glycoprotein. So P-glycoprotein, it is located into the placenta, located into the GIT, located into the brain, which is important for the efflux of the drug. So when you take the drug, there is a special pump which tends to eject the drug to prevent the absorption. So this efflux mechanism, it involves the utilization of the ATP directly. So it will be hydrolyzed, releasing energy that will reject the drug. So see this diagram here, it indicates especially for the antineoplastic drug, or we can see now indeed in case of the cancer cell. So cancer cell, it has a tendency to develop the resistance to the most of antineoplastic drug. So how does it provide this resistance? It has a tendency to develop this P-glycoprotein pump, which has a tendency to efflux the drug. So when you take the antineoplastic drug, the pump has a tendency to efflux the drug, means to pump out the drug so that to prevent the access of the drug into the cancer cell. But if you see now how this pumping is happening, in this case, ATP is needed so that the drug will be pumped out through this P-glycoprotein. So for the successful mechanism of the antineoplastic agent, for this case, you have to take the P-glycoprotein inhibitor. So if you take with the P-glycoprotein, it will inhibit this pumping mechanism, whereby the antineoplastic will get access into the cancer cell and kill those and prevent the this unprogrammed cell growth. So the substance like the grapefruit, this acts as an inhibitor of the P-glycoprotein. Secondary active transporter, as you say, the energy is not derived from the directly from the ATP, but it is derived from another substance. So as usual for this one here, it is utilizes the solute carrier transporter, which is important also for the efflux and influx. So as we have discussed it for the case of the symport and antiport. So see this diagram, which involves the carrier transporter, but active, which involve the utilization of the ATP secondary active transport. For example, the transportation of the glucose and the amino acid in the small intestine, and in case of the small intestine and the ileum or the jejunum. So here glucose is very important to be transported across the carrier matrix, which is the solute glucose, the sodium glucose transporter, which involves the sodium glucose transporter 1 and the sodium glucose transporter 2. So in this case here, the diagram, it indicates the symport transportation of the drug. So this is the symport. So for this case here, as you can see, this is the jejunum and the ileum, the absorption of the both glucose and the amino acid is taking place through the secondary active transport. So we have seen those carrier transport, which is the facilitated and active transport. Now let's see the pinocytosis, which is the last method for the transportation of the drug. So this involves the engulfing of the drug across the biology, engulf the drug and put into the synaptic vesicle, so special vesicle that the drug will be ingested. Then that vesicle, it will transport across the biological membrane and get released into the another side of the biological membrane. So it's not necessarily that the drug which is transported through this pinocytosis has to be released into the another side. There must be possible for the trapping and forming the vesicle, whereby the drug will be stored into the body, or it will be released inside of the cell, or it can be released into another side of the cell. See now this mechanism here. So this, it indicates this is the biological membrane, whereby the drug bind to the special binding site, whereby the membrane, it will make the folding and enclose the drug, forming the vesicle, which will be transported to another side of the drug. So this is the same mechanism which is taking place. For example, the albumin bind on the surface of the plasma. Once it binds on the plasma here, you find that it will form the vesicle. So this vesicle, it will be transported across the biological membrane and released into another side of the cell. So it's very important for the transportation of the drug, which is considered as the huge molecule. For example, the insulin across the blood-brain barrier, vitamin B in the in the intestine, and the norepinephrine as a neurotransmitter, and the polio vaccine. These are the huge molecules whereby the pinocytosis mechanism is very important to transfer the drug across the biological membrane.